today, we'll be able to remember what we discussed last time as to genome prediction, genomic selection, how it's being used, and all that. And then we'll look at how the data are being prepared. How do you select a model? Because if you remember last time, we talked about the fact that your data is very important, how clean they are, both genotype and phenotype, and model selection, depending on the traits, it could affect your accuracy. And then we'll see the demonstration in R. We'll look at the results, how it's being interpreted, and how do you use those results um, for example, genome selection. So, sort of recap, we know that genome prediction is actually it's a statistical approach. So it's an approach why the genomic selection is when you implement prediction into practical aspects, right? So it is just using your DNA markers to predict the breeding value or genetic merit of an individual or genotype, right? And then the importance, as we said, is, the ultimate goal of breeder is to increase genetic gain, and that can be done by selection, increasing selection accuracy, increasing the intensity of cell selection, and also if you have a way to reduce generation interval, this will cumulate to increase in genetic gain. So now we're talking about data preparation. So if we have read data, for example, you have an experiment that is set out, and then you collect data depending on the on how the experiment is set. We have different design. We have our random and complete design. We have alpha design and the likes. So sometimes you need to do spatial analysis to be able to get your blues or blob, as the case may be. I can't stress this enough because sometimes some people are not really take time to analyze their phenotype from maybe making sure that they take away environmental effect or probably the experimental design error from their phenotype, and that can potentially affect your prediction. And same as your genotype, genotypic data, you want to make sure that it's clean. You want to make sure that you apply quality control, which I don't want to go into details of that because that is maybe not the focus of this webinar. So your data can be in different formats. It could be in APBA, it could be in VCF format, and it could be in text file. And then your phenotype, depending on what you are doing. But the most important part is that your phenotype in your genetic data must match your phenotype data. So you must be very sure that this is matching. If it's not matching, you're going to have problems in your analysis. So I think for us, I don't know, can I share the script in the chats box?
Yes, please, you can. You can.
Oh, okay. Okay. 1 sec. Please kindly go ahead and download the scripts. Let me put the rest so that we just won't have a reason to stop again. Do you have access to the first one now, please? Someone should confirm so that I know that we are. So I sent 3 scripts. Do you have access to them?
You have shared 3 scripts so far. I see them in the chat.
Yes, yes. Please download the three scripts and then make sure you save them in the same folder, and then you can open the first, the one with simulation, so that we can get phenotype and genotype data. Are we together? I don't know how we're going to know if people are following in this kind of settings.
Hi colleagues. Please, if you are able to download the scripts, it will be great to show by hand so that Dr. Akinade
Yes,
is able to know that we are all on the same page. So if you are able to download the script, kindly just show by hand so that we can know what to
thank
do.
you.
If there are any challenges
Uh,
as well, yeah, so you can even give a thumbs up in the chat as well when you have been able to download the script, or show by hand, and we can see that as well. If there are any challenges, to please let us know in the chat as well. Thank you.
yeah, that's perfect. So let's know. Let's download the scripts. And like I said, just save all the three scripts in one folder. So that, and then once you've done that, you can open the one that has with simulation, so that we can simulate our genotype and phenotype. I mean, this is what you have if you're doing the real analysis experiments. You will have had your genotype, you have your phenotypic data, and then if it is raw, like I said, you need to adjust them. And then once you have your phenotype and genotype, which we're going to improvise because I think that would be the best way to handle this kind of webinar. So we'll just wait for us to have significant number of people who have been able to do it, so that we can move at the same pace.
So Dr. Akinade, Enoch says he's been
Yes.
able to download, but he cannot open. I don't know if anybody is facing a similar challenge.
Let's know if anybody is successful. Um,
Okay, so yeah. So Enoch, kindly open it from the R Studio, because if you already have R Studio, once you open it,
oh,
the R Studio,
I
you
just oh thank you. One person is successful. So what I was thinking maybe I passworded it or something. So yeah. So
so
maybe the person.
I myself have been able to open it as well. So kindly,
Okay.
if you have R Studio, you should be able to open the scripts.
So President, just tell me when you think we should proceed. Probably the number of people who are successful or something, and how you want it to. You will help me a lot. Sorry, so
Kind,
that you know.
I think we can proceed. I think we can proceed because a couple of people have been successful. So I think we can proceed.
Okay, so let's proceed. So I will share my screen. The screen. Can you see my screen? Our studio screen.
To follow up this, Damaris.
Yes, we can see your screen.
Okay, so if you
Wait and see.
actually install, if you have installed RR block, you can run the first junk. So this is the package we'll be using for this simulate. I want to simulate the data. And this is just for you to be able to reproduce the same thing. Let's say you want to rerun it next time. And then here we are saying we want to have individual or genotype number to be 1 to 10. You could use 200, you can use 300. It's like you are having your population size or your number of genotype is 210, and then you want to use 5,000 marker. I mean, so that is what we want to use here. And the reason why I use 5,000, it could be more than that, especially if you are using sequencing data or WGS data. But for us to run it faster, so I don't want to have too much big data, because it is the concept that we need to get so that we can play with it on our own time, at our own spare time. So now we want to actually generate,
Dr. Akinlade.
Yes.
Sorry, Mary has her hand raised. I don't know if she has a question or she's struggling with something.
Yeah, sorry.
Okay.
Thank you. Dr. Akinlade, I would like to know these individuals. Can you describe them, please? In terms of diversity, how are they diverse? How diverse are they? What kind of population?
Okay. So here we are, we are just simulating it. It's not like one particular population. It could be any population, depending on what you are researching. Here we don't have specific population. We don't have it's a breeding population, germplasm, or diversity. No, we're just trying to say we have at 210 individuals. So did you get that? Like, we're just simulating a population to work with this kind of.
Can you advise what works for prediction? Can you advise? For one gain, right? So genetic gain and all that. So can you advise?
Okay, it depends on what your prediction is aimed to achieve. For example, if you want to use it to select best variety, for example, if you have a magic population, do you know what the magic population is? So you have 8 parents, and then you cross them in different combination, and then you have your real line from magic population. You can use prediction to select the variety in such a magic population. I know you can also use genome prediction to select parents for your crossing work. For example, if you have diverse panel, and then you want to see, maybe you want to pick the best 5 parents, you can do that. So depending on your objective, that's what will determine the population you're going to use. But basically what you need is a phenotype and a genotype of the same individual that you want to use. Like, if you are part of the webinar last time, we talked about the population that is used, and we talked about reference population that it has to be genotype and phenotype. So we're just improvising, so you don't have any population in mind. I hope that's clear.
Yes, sir.
Good.
Thank you.
No worries. So here, what we are just trying to do, if it is a read data, we don't have to do this, because we know that in read data, some of the SNPs will have true effects, and some of them will not have any effects, depending on some of them will have effect and some of them will have. So I just want to assume that out of the 5,000 that we have in mind, let's assume that 500 of those markers have true effect. You can change this number as you like, because what is really important in this is just to see how it really works in reality, right? So you can change it doesn't change nothing. And I hope we are also running it so that we can get phenotype and genotype to work with later. And here we have a genotype that assume that 500 of the 5,000 marker are true effects. Like I said, you can play with this number; it doesn't change anything. So now you have phenotype, you have genotype, and of course, let's save it in the same folder. So here is just to preview the data that we have. So, this one would be if it's real data, this would be your genotype name. For example, I work with GP, that would be ICC stuff, and I don't know which crop we work with. So this will be the normal genotype name. But here is just individual 1 to 210. And then for the phenotype is individual 1 to 210, and then it assumes that whatever trait it is, let's assume that this is yield, which I mean it's not going to be yield because this is negative. So whatever trait it is, right? Each individual will have one value, and then you have your genotype. I hope you have all done that. And then we have, if you check the folder, you should have your phenotype and genotype saved in Excel. Is that correct? Chat. What do you? Let me know if you've been able to save the phenotype and genotype data file. Okay. I see one thumbs up. Done. Okay. Okay. Good. Let's move on. So I don't know. So now we have our data, which you get if you do if you have real data, and so now you have phenotype, you have phenotype. What then next is to select the method, model method. I'm going to use our block, and I will use one Bayesian method. And like I said last time we met, what is different between models are assumption. So for example, in Rara block, it assumes that all the SNPs that you put into the model have effect that is small, but not zero, right? And depending on the Bayesian that you're using, they have different distribution. So you could have the zero. Those one assume that there could be zero. One of my favorite model is this HAR that I work with when I was doing some complex traits, because that assumes that he assumed that your SNPs have different effects, some with big effects, some with medium effects, and some with zero effects. So you can have mixture kind of effects. But we're going to play with R blob, which is also known as, I mean, depending on if R blob or G blob, if you have pedigree information, you can apply it here. But it's because it's simple to run, and then it's actually being used for different crops. So we're going to use our blob and the C to run this analysis. So if you go to the scripts, you can open the one that has R blob. Are we there? Yes. I'm not seeing thumbs up. Have you opened the script that has Arab block? Okay, one yes. Please, can someone share script?
Please let's respond if we've been able to open the second
I
script.
think someone wants you to reshare the script. Oh,
It's been reshared. It's been reshared. And yes, Doc, we can go on. People have been able to. Ogechi,
yeah, thank you.
to reshare. Can someone reshare the scripts? Okay.
So let's load the packages that we need for this. You must have installed them already. So it's been given to you. So now, the simulation is because we don't have real data. If you have real data, this is where you start your work. I mean, assuming that you have adjusted your phenotype, you have cleaned your genotype. So let's load our files. Genotype. So we can open it. So this contains 410 individuals. And then 5,000 markers. So marker, depending on what you're working with, it could be, but here we just call it SNPs, right? So it is individual by SNPs. And then if you check the phenotype, you have two columns. You have individual and you have phenotype values. So this will be your real trait values for the for all the individuals. So this step 3 is very important because sometimes if you don't check this, you might end up having issues and then you don't know where that comes from. So you try to match phenotype and genotype by the individuals and then see and be sure that you have every individual carrying the same, individual from phenotype is matched with individual from the genotypic data. Let's open it and see. So you see, now you have merged the genotype and phenotypic data. So individual 1, the phenotype is this, and the remaining columns are the markers. Hope I'm not too fast, and then we are following, please. Are we following, please? I need yes. 1 yes is fine. Okay, thank you. So after that, let's do you remember I was talking about gene matrix, genetic relation matrix, where you look information from you use the information shared by individuals to look at the relationship between them. It's more or less like what you do by looking at correlation between two individuals. So, but you are looking at their relationship based on the shared SNP here. So if you run the junk 4, you're going to have our gene matrix created. And this is what gene matrix looks like. So the pair of individuals. So you are looking at what they shared, what individual pairs of individuals shared based on the marker data. So this function is used to calculate your genetic merit or breeding value. So don't forget, like I said, this is just to be able to get the same result if you rerun it. So when you run like this, you are sure that if you rerun it, because sometimes if you are, for example, if you are working with methodology paper, you want to use some of the simulated data, and then you want to make sure that if you come back to run it, you get the same thing. So that's what this is for. And you run this, you have to.
Hello, sir. Can you repeat that, please?
Oh,
What you
you
just
mean?
said?
So I said this test seed is usually applied in scripts to be able to have to be able to rerun the script and get the same value that you got initially. We are using simulated data. Let's assume that I want to um, publish a methodology paper, and then I didn't have real data, and I simulated it. If I don't put this, if I'm running it again, and I'll be getting different values. But to prevent that, that's why you put this. So what I'm trying to say is that you don't have to put it if you have your data. So it's not doing anything. It's not like it's helping you to do any prediction. Is that clear? So what you need to get your prediction is this function.
Yeah,
Okay,
so
thank
it's
you.
the mix.solve I'm asking about. The mix.solve.
Oh, that is the function from RR block to calculate your GEBV. So
Okay,
it's going to
thank
give
you.
you different parameters, and GEBV is one of them. But we just go and have so if you look at it, we already have GP model. So if you open GP model, so you see you have VU, we have VE, environment, beta, and then U. The U here, if you remember when I was talking about ridge regression, where you have lambda, so U here is actually a GBV. It's your GBV, and you see that all your individual we have genomic estimated breeding values, right? So because your model has this different parameters, you can then just get out this U. So just to get the U, the model $U will give us the GBV that we're looking at. And then if you look at just the first few individuals, this is their genomic estimated breeding value. Now, so if you want to get genomic estimated breeding value, and you are using RR block, you've gotten it. And then if you look, what can I do to do something? So if we open, this is it, right? If you open this, let's just save it. Right. Can you see what I'm doing?
Yes, we can.
So if you save it, you can have the file saved for you in your folder. And then you can see from individual 1 to 210 have their estimated value. That is the GEBV. However, I mentioned about accuracy, knowing the accuracy of prediction, because yes, I will touch on it. Somebody was asking how do we interpret GEBV. Yes, I will talk on it, but yeah, later. So I'll touch on it later, please. So now I have prediction. The question is, how accurate is my prediction? And I mentioned it the last time. I said there are different ways, I mean, a few ways that we can use to check the accuracy of our prediction. Accuracy of prediction is saying how accurately you're able to predict them using only the SNP marker. Don't forget they have phenotype, but now you are using only the marker to predict their effect, and then you are asking how accurate is this prediction. Again, this is the prediction already, but you want to see how. And I mentioned that we can use cross-validation method. We can use leave one out method. We can use CV1, where if you have multi-environments, you can use one environment to do the other environment. I mentioned that last time. So, but for the sake of this, we're going to use five-fold cross-validation. So what does five-fold cross-validation means? It means, let's say now that we have 200, you divide it into 5. You use 80% to develop the prediction and test your prediction on the remaining 20%. That's what this will do. So let's give it a run. So let's see what is the accuracy. Accuracy is 0.118. 0.118. What does it mean? It means you are able to you're able to produce just 11% prediction. It's like correlation, right? 0.11%, 11%. So this is very low, right? Don't forget that we just simulated data. However, you can get something like this in yield. Yeah, I think I got something like this in yield before, because it is very complex and then it's very hard to predict. So, but you can also so the question you ask is, if you want to predict a trait that is very low, because it's very complex. The complexity, if you remember, when I was talking about accuracy of prediction, I was talking about different factors. I was talking about the density of marker can affect it, the heritability of the trait you are using, and some of these things. And we know that yield is very complex. So what you can do is to use multi-traits. You can use trait that is correlated to yield to actually increase the accuracy. But I'm not going to be touching that in this webinar, but that is how to actually work around some traits that are really very complex and have. Another thing is you could try different models, because what this is telling you might be that this model that we use, the assumption is that they assume that all the SNPs that you feed into the model actually have effects that is very small. But if in reality, some of your SNP actually have big effects, this model is not the best, because it's not able to actually capture what is actually available in the data. So sometimes you need to use maybe one or two different model to check the best one. And that is based on the assumptions that the model have. So, but yeah, like I said, you can also use leave one out. Leave one out is, for example, you have 210. What it does is that it uses the first one as the prediction, as part of the prediction for the model, and then take out second one to test for the prediction. Fourth one, prediction. Fourth one, useful to test the prediction. So that's how this one works. This is the five-fold is dividing the entire population into 5. Use the 4, which is 80%, to predict and test the prediction on the 20%. Leave one out, actually divide into half by taking the first one as the prediction population, the second one to test the prediction, which means it's going to divide into 50-50. So it's more stringent, if you ask me. And then I mean, this will take a longer time than the first one because it's actually taking one and leaving one out. They're taking one and leaving one out for um, validation. So, well, like I said, don't mind this correlation because it's not real data. So this is just low accuracy. So this is actually telling you that you are not predicting more than 11% accurately. And of course, that is very low, if you ask me. Let me share my PowerPoint again because I think I mentioned what is. Okay. So yeah, I said typically you have 0.3% to 80%, like 3%, 30% to 80% most traits. You should be able to predict it. Even yield some, yeah, you should be able to predict 30%. And for traits that are highly heritable, you could have even um, 80, 85% accuracy. So the accuracy is actually your phenotype by GEBV. What that means is, for example, for 5-fold calibration, you're going to use the 20% phenotype, command the EBV of those 20%, and then that will give you accuracy. And for leave one out, you're going to take the 50% that is taken out to confirm, to use to check the accuracy, and you correlate it with the GBP of those 50%, and that will give you the accuracy. Okay, somebody was asking, how do we interpret the results? So let's do it quickly. So you rank the individual. So let's open the GBV that was saved. GBV. So depending, okay. So are we here? Have we opened the GBV Arab block? Yes. Okay. I have to go and open the chart to see. So now you rank them based on their value. So it's saying that individual 193 is having the highest genomic estimated breeding value. So this is based on their genetic merits. 193 is the best. So let's say you want to select best 5. It means you're going to go with individual 193, 74, 178, 50, and 208. Let's say you want to select best 5, best 2. This is how to use your genomic estimated value. So I don't know. Is that clear, or I should explain further in terms of how to use it for selection? Hello?
So Enoch has his hands up. Enoch, is it a question or is to agree that it is clear?
Yes, I would like to ask, in this, what he's talking about now is these are the values. For example, he picked number 193
Mhm.
is the one with the highest GBV.
Estimated,
So,
yes.
yes. So now I'm looking at for different trades now. You have
Yeah.
this for one trade and you have a different value for another trade. So from now a breeding perspective, now I understand, yes, you may have targets for different trades, but now
Yes.
overall, how then do you put them together? How do you use them?
You mean if you have multiple traits?
Yes.
So now, if you have multiple traits, of course, in breeding, that's one of the challenges. You want to combine two, three different traits. If these traits are not highly correlated, you know what it means. If you have two traits that are not highly correlated, that means if you are selecting for one to increase, the other will be decreasing. For example, if they are negative correlated. So this also comes to play in this kind of work. So you have to, if, for example, if if they are correlated but they are not strongly correlated, maybe you don't want to pick the best of the first one. You want to pick maybe um um something that is not the best but it's not the worst for one trait, depending on the traits you are combining. So this is what actually has to do with you, the breeder, you know. So the science part and then the art part as a breeder. I don't really get it. So it depends on the relationship between two or the three traits that you're looking at. And don't forget, um, also depending on the traits that you're looking at and the understanding of what you're looking at. Maybe the best GBV is not what you want to use for certain thing. You just want to have at least um, a very good kind of genotype. But if you are looking for example, if you want to select just an example, highly tolerant, maybe you pick 197, and then if you want to pick the worst, you're going to pick the last one. You get my point? So it depends on the objective. So it's not like you have to pick. I'm just saying if you want to pick the best five, it should be the best five when you rank them. But if you want the worst and the best, also you can pick it from the estimated mean value. Again, don't forget that the reason for genomic selection is to be able to select on genotype individual by you using a population, the same population that is genotype and phenotype, so that by the time you you want to select, then let's say you want to go to the second generation, you don't have to wait for the phenotype. Just by, and don't forget that our prediction actually estimated the effect of each of the SNPs, and then you use that prediction model to select another population that is not yet phenotype, but and you use it to select them. So that is where genomic selection is useful. It's not useful when you have phenotype and genotype.
Okay, thank you.
Yeah. So, like I said, it depends on the application. If you want to select early material, early population screening, parent selection, that will determine what you do. And if it is multi-trait, you have to analyze the trait and have GBV for each of the traits, and then select based on that. You can also, if you
Hello, Doc. Sorry to
yes,
cut you. Mary has her hand up. I don't know if she has a question.
Yes,
go ahead,
thank
please.
you. So this last comment you just made, about when it gets to the second generation, you know. So my question is, are you going to do another genomic prediction, or you are going to, as you say, use the genetic information or genotype information of the former population, the training population, right? So,
Yeah.
but now we want to align it with new individuals.
Not new individual. This
Okay.
individual are part of, for you to do genomic prediction successfully, the individual that you want to select, let's say you have 200, you use this 200 as just in your mind, assume that they are really individual and their name is individual 1 to individual 210, right? So I
Yes, sir.
used individual 210, that is phenotype and genotype, develop a prediction model, right? And then let's say I pick 50 of them of the same individual. Then I plant it. And then after a few days, I cut the leaves and I genotype them. Based on that genotype, I can get the genomic estimated breeding value without, for example, if I want to select for yield, and I already predicted the 210 individual for which they have yield, phenotype, and genotype, and then develop a prediction model. If I now have the genotype only from the individuals, I'll be able to select them based on genetic estimated breeding value, based on their DNA. I don't need to wait until the plant grows in the field, mature, and then harvest. I don't need to wait. But you cannot pick population outside the population you used for prediction. It won't work. In fact, the the, the closer the population you want to apply genomic prediction to the population you use to develop the prediction model, the more accuracy you're going to get.
Okay, so you now take the best 5 and you make crosses among themselves, and you now have, real, and again, you're starting all over again. Or best 5 and worst 5. Maybe those worst 5 have other traits that are important to, your
Yeah, I
program.
mean, that depends on what you want to do. If, for example, if it is to select maybe plant material or you want to just screen your population, that's another thing. For example, if I have repopulation after series of crossing, I develop a magic population and I want to do trial testing, I can just use genome prediction after the first evaluation of phenotype and then develop a prediction model. I can then, each time I plant them, you want to do a few number of samples to really test them across, and then you can use that to just be selecting based on the prediction that I've made before. So it just depends on the program you are, the objective. But what you should have in mind is that the relationship between the population that you want to use your prediction on and the and the and the breeding population should not be too distantly related. The more closer, the more related they are, the better your prediction. You get my point?
Yeah, but I thought, sir, that when you have done the prediction, so in
Yeah.
future, you're not using that population to do things. In future, you want to, fall back on this without having to phenotype again. I know.
Yes.
But you're going to have new individuals, kind of. Those ones that don't have
But
genotypic information,
so
you're going to have new individuals. So how do you match them with when you don't have phenotypic data? You generated matched, you merged phenotype and genotype. How do you bring
so
individual without?
one thing is you use the same genotype. You genotype them using the same genotype platform. So for example, if they are genotyped with, let's say, 5K SNPs, this new one is also genotyped with the 5K SNP. So, and you have the effect of each of the 5,000 SNPs from prediction that were made before. So based on what they now have from the genotype, that's what you sum to get their GBV. GBV is just this sum total of the effect of the marker that you have in the prediction.
Okay, not the phenotype.
No. So
Okay, sir. So
so
what you're saying is that I can keep phenotyping without genotyping. Is that what we're saying now? You can keep
no, you
phenotyping others.
genotype. No,
Okay.
you genotype. It is the phenotype that you don't need at this point.
Okay.
Because phenotype
Okay.
take time, right? The the biggest part of this stuff is to be able to select more, increase selection intensity, to be able to select fast. If you are, for example, if you are looking at, let's say, yield, if you have to wait to plant, and then to mature, and then to harvest. That's when you're going to see your yield, right? But because you want to reduce that time, that's why you already estimated the effects of the SNPs of the marker that you have in that population, and then you genotype the new population with the same marker. And because you already have the effects, SNP effect of all the marker, you can then look at the marker that they carry after the genotype and then sum it together and have genomic prediction value for the new one that you have, and then you select them based on that. So you don't need to have phenotype for them.
Okay, thank you so much, sir, for the clarification.
Thank you. Thank you. So, do you still have time? So yeah,
Hello, Doc.
best
There are some two questions in the chat box which I think you can address. Doc.
Thank you for.
Okay.
Yeah. I'm trying to check the chart now. You use one-step approach, two-step approach. So when it comes to methodology, two-step, one-step, like it depends on the type of data that you're using, right? So the R blob it has done it for you. You don't have to separate. The two-step approach is there when you have SNP separately and then you do the second step. And then when you also use ASREMO. So these are just methodologies. It is not the. So what I try to do is just to show how your GBV is calculated and then how you can apply it. But you can use different methods depending on your data, your traits, your understanding of the trait. If the traits, for example, if you have idea that the trait is already having like significant KTL, you know where they are. You can actually include those KTL as a fixed effect to improve your accuracy. Those are the ways to actually increase the accuracy of your prediction. And what is the accuracy of prediction? It is ability to predict your traits better, right? So those are just methodology on how to increase your accuracy, the accuracy of prediction. But the real thing we're talking about is this is how to predict them when you have genotype data and phenotype data. So do you develop model? I can see. No. Are you referring to the person that asked the question? So the developed model can be used for another population. Another population that is not related to the population you use to develop prediction model, not model itself, prediction model. It will, if you use it, you have nothing, accuracy will be so low that it will be useless. So you want to use population that is actually close to the one you want to use it on. So that's the answer to that. Because if you apply it, the accuracy will be so low, and then it will be useless doing such activities. Yeah, I think someone is raising up a hand, or is Paulina? Yes.
Hi, Johnson.
Hello.
Thank you so much for the wonderful presentation. My question is, assuming I phenotyped my population, and after estimating the breeding value, I decided to choose 5 of the best as my parents to generate magic lines. The first phenotypic data I got was done at location A. And now I want to be able to see how they'll perform at location B. Would you advise I start the whole thing, the whole process, and then phenotype this magic population at location B, or you suggest the phenotype data I'd already gotten from location A might be enough to estimate their true genetic value? Thank you.
Yeah, Paulina, thank you. This is a very good question. So that depends on on some factors. One of it is the heritability of the trait you are talking about. Is it highly heritable? Of course, if it's highly heritable, it means that it has um, low influence of environment, right? So if
Yeah.
that is the case, you can. However, I don't forget that. So because you are moving from one environment to another, if your trait is very complex, it will not be sufficient to do, because what then happens is that your accuracy is going to be reduced. Because what determines accuracy, if you're part of the last webinar, we talked about accuracy as being affected by irritability. And then if you have a trait that is really, really affected by environment, that will reduce it. And then that will reduce accuracy. And then when accuracy is reduced, your work is not there's no assurance. I mean, you are not confident in what you are predicting. That's why I said when I showed genomic estimating, I said these are the values. But you want to also question further to say how confident is this prediction? That's when you go to check the accuracy, right?
Yeah.
So if the trait is affected by environment, definitely that will have reduced your accuracy. And then depending on what accuracy you have before, you then ask yourself, do I have confidence in using the prediction model? So most times, people that use it in companies, because they already have pipelines, they have they have their pipeline companies where they use some of this method. So if it is not a different environment, they have different prediction for different kind of pipeline. And that's why it's more useful for people who use it commercially in companies who they have control over their population that they use, the parent that they use every now and then to develop their materials and all that. So it's good for them. But in that case, it depends on the traits. And more often than not, you don't have the accuracy that you have again, because that's already affected by your other factor that will have affected your model. So you might end up needing to do your prediction again, because accuracy is more important, because either it's accurate or not, you have your GVV, right?
Yeah.
But if it's not really accurate, what accuracy, let me repeat it again. Accuracy is talking about how confident how confident are you predicting this? How close is this value to the real value? So if you have accuracy of 80%, it's better than accuracy of 5%.
Yeah.
What affects accuracy? Environment affects accuracy, data, and then your heritability. And then when you change environment, that's one of the parameters that changes with population.
Okay.
So because it's affected by environment. So that would be the case. And again, for genome prediction, it has to do with, if you want to implement it, genome selection, using um packages to do it is it's not simple, but it's easy. You can combine different things. I can even fix gene by heat into the model, complex model, especially if I'm doing it in X-ray mode. That's possible. However, you have to also know the practical aspect. You have to be an experienced breeder. You know your crop. You know some of the factors that affect some of your for example, when you plant it on the field, you know factor that affect it. If you can actually, fix some of this factor in your model, you can also have good prediction accuracy.
Okay. Thank you. Just a recap, if I understood you well. So that means, let's say in Ghana we have 5 different locations, and I am really sure that 2 of the locations really have similar climatic conditions and soil other not conditions. And I have observed that over a long period of time, I know that there's really no vast difference between these two locations. That now means that such a prediction can easily be used for the second environment because of they having similar conditions.
Yes.
But in a situation where the environment is just totally contrast to whatever I had used a model initially, then I have to be careful and don't even consider because, okay, all right.
Yes, because of course, you know that if you even go to the field to plant to get the phenotype, if you don't, the reason why you're using genomic prediction or genomic selection is to take away the time taking to plant this and that. You know that if these two environments is very different, if you plant them, your phenotype will be very different too.
Yeah.
It will be very, very different. So that's whatever will affect your phenotype significantly, we also affect your prediction because your prediction is actually predicted by environment, by phenotype and the genotype. And what you do mostly is to get your SNP effects, the effect of SNP that you put into the model.
Okay, thank you so much.
You're welcome. Then we can say, prediction focuses on estimation, estimated potential. Yeah. Yeah. So, Femi, this is correct. I'm trying to. Can you still see my PowerPoint? Hello. Hello, can you still hear me?
Yes, we can hear you.
Okay. So, um, I think, yeah, I've come to the end of my slide. So, general prediction improve reading efficiency. The techniques is around the fact that you want to increase. If you can do this faster, then going back to breeder's equation, you can reduce the time and then you have tendency of increasing your genetic gain faster. And then Arab blob has been used widely for different crop and also different traits. But in my experience, there are traits that I've worked with that have to use Pishar because it works better, especially if you really know that trait. Let's say you've done GWAS on that trait before and you know that there are significant, regions that is actually, so you can have significant effects. You can actually use our vision house for such a kind of prediction, and that will give you a better accuracy. So again, you can always overemphasize the fact that accuracy actually, the more you are able to, the better the accuracy of your model, the better your selection will be. So don't just estimate breeding value and then just select your plant. You have to know also the accuracy of the model that you have developed. So that accuracy is what you bank on for your selection, and the better it is, the better the accuracy, the better your selection. Yeah, I mean, we're gonna stop there.
Okay, so I think the floor is now open for more questions. So if there are questions, Mary, you can unmute and ask your question.
Yeah, thank you, Doc, for the time. I'm really grateful. But Mr. Joy in the chat, she mentioned something about phenotyping across different environments, then averaging, doing an average before you go into the GP, genome. So is that a good way to solve diverse environments?
I think that genotype to be used for the prediction should have been tested in different environments and the average performance used to have.
Then
Okay.
lastly, you didn't touch on base C. The base C,
Oh,
yeah.
okay. Okay. So, oh, yeah. What should I do first? Should we do base C
Please
or
answer the
I
question
should?
of Joy, please.
Okay. Okay. So this is a very good one, and this is around your understanding about how to manage environmental factor that influences environmental factors that influence your trait of interest, right? So if you test your phenotype across multiple environments and then average them. So what you are trying to do is to reduce environmental effects, right? To say, okay, I've captured the environmental effect. That can help. However, most of the time, it's not the best. What I would do in this case is um, doing spatial analysis where I'm going to fix some of this location. I'll fix location. I'm not going to just get phenotype from A, B, C, and then average them. Rather, I'll do spatial analysis and then fix location, and then I'm going to have blues. That adjustment is much, much better. I don't know if Joy understands. So you are not going to just take phenotype from A, B, and then add it together and they take average. I think people do that, but I think that's not the best way to do it. You do spatial analysis where you because you already um tested the same genotype across environment, then you can use environment as this effect, and then you can get phenotype for all the directions, all the environment. That can actually increase your accuracy. So let's open the last script. Is the last script that has let me see. So before we run that last script, let me just tell you, we are just trying to use, it's like, let's say I want to do genomic prediction. I don't have idea of the trait. Then I use R blob. I want to see if this C will be better, you know. And how do I know it's better? It's by checking the accuracy. So let's try and run it. Can you see my R Studio? Can somebody talk because I
Not
don't
yet,
see
sir.
chat.
Not yet, sir. But why are you choosing base C? What of A and B?
You can use it. You can use B. You can see. So there are packages that you can use to run C. Okay. With with BG, what's the name of the package you are using here? Hold on. Sorry, let me. With BGLR, you can run base C, base A. So I'll show you where you can change the model. I just use C just to demonstrate. You can use A, you can use Lasso. These are just different model with different assumption. And based on the assumption, they can give you better accuracy. Is that clear?
Very clear, sir. Thank you, sir.
So let's run the last one. I don't. So let's open the one that has base C. That one we can use it for base A, base B also. Okay. So you can repeat it, but we already loaded the same file. So you can start from step 4. You just want to see this model because it's same genotype and phenotype. We just want to see if the C is better for this trait. You see, if you go to step 6, you see where I put base C. That's maybe that's taxing why if you can use it for. So if you put base A, it will run, and then it will be model for base A. If you put B, it will run. So you can actually check to see the difference between all this model. It's just around the assumption, and based on that assumption, they perform better in some, depending on the traits that you are looking at. And it's better that you at least use the best traits, the best model for your best trait. I don't going for blobs or blue, for example. What I don't get that question. Going for blobs or blue. Is it
That's
that
in
you're
relation to the
asking?
multi-environment, averaging over multi-environment. He was trying to suggest, I think.
Yes, it
Can we
was,
have
it
that
was
person?
about the multi-environment phenotypes, so that estimating the blobs or blues can get us better estimates, so that it is better to use them when doing the prediction. I was just trying to comment on that. Yes.
Oh, okay. Okay. Of course, we know the difference between blobs and blue and how you can get them sorted. So yeah, Mary, this is where you change it. If you change it to A, it will give you genome prediction based on patient A. So if you run this, I think this take a little bit of time because it's going to run iteration of 1200, 12,000 and points. I'm trying not to go into the model specifically. So I'm just trying to show how to predict genome prediction. That's why I'm not going to 1 step, 2 steps, and then different model. Don't forget, it's just introduction. I don't want to make it too complex. That will not get the real aim of the webinar. So again, you can run this one yourself at your leisure, check the accuracy, compare it with the one you have with RBlock. Most time, if the trait is really complex, maybe there's no significant marker effects. All of them have, infinitesimal kind of model assumption. All this model will perform the same way. There will not be different in accuracy. The only time you have different accuracy is if the assumption of one model is actually more fitted for a certain trait. So that's when you have different accuracy, and then you are expected to go for the best accuracy at least. That's why if you take papers that has to do genome prediction, maybe in the past few years, they usually compare different models to say, yeah, having you done this model perform better for this trait, this model perform better for this trait. But we have gone beyond that now, that we can now, at least for some of the major crop, we know what model will perform better for what trait because several research has been done. But now we are at the point at which we want to improve the accuracy. And how do you improve accuracy? I mentioned some of them last time. So different things, anything that you can do to improve your phenotype that you want to use for prediction, and you can use to improve the quality of your phenotype and also quality of your genotype, will eventually help you to increase the accuracy. Is it possible to use all the model then compare? Yeah. Exactly. Especially if you are working with a new material that you don't have nobody that's done it printed in the future. Yes. If you want to do genomic selection, you have to be sure that the model you are using gives you the best. Yes. Yes. Don't forget, the more accurate you're able to predict, the better your selection. So yeah. And that's why you see, even some companies that started implementing genome selection, they will not use it for certain traits if the accuracy is not high. But now we are trying to use AI um to improve to improve the accuracy and all that. So that is what's also ongoing. So it's all about how best you can predict this genotype. So yeah. Again, with this now, all we're going to get is the C model prediction, and then we will get GBV from the model. But we have to go one step further to find the accuracy of that model, which is the code. Okay, it's still running. So if there's no other question, I think we can call it a meeting. President, is that fine?
Yes, please. Please, are there any more questions or everybody is fine? I don't see any questions in the chat. But are there any more questions or we are fine? Please, if we are fine, we can give a thumbs up in the chat. Can we have some thumbs up? Very good. A couple more would be good. Maybe before we leave, Doc, we'll be grateful if you can share your email address in the chat so that if anybody wants to reach you for further engagement, they could easily do that. So we'll be grateful if you could share your email address in the chat.
Yeah, that's totally fine. Yes.
So Doc is going to share his email in the chat so that we can reach out to him. Let's not overwhelm. Well, somebody has a question, even as we are wrapping up, and it's from Abdul Salau.
In my opinion, you can treat it like you treat GWAS. You want to have at least significant number, because if you want to, don't forget that what you are doing prediction for. Okay, how do I handle this? Let me do this way. Don't forget that what you are doing, except if you are asking based on you are asking the question for their own prediction paper where they just predict this and that and that. But if you are if you have the mind of if you are doing it to predict and then you do for selection, of course you have to have population that you want to really check what is in there, right? So um better population increase if you have big size, it will help you to really be able to trust what you are doing, right? But because here you are not doing you are not looking at association between one SNP, right? It is you are fixing the SNPs together, and then you are looking for the effect of each of the SNPs. So uh, my take would be that I mean sizable number will be more what's the word now? It will give you sort of confidence, right?
Okay, please.
Thank you. I love your I don't know how to pronounce your name. Is it Abdul? Thank you for everybody's. I love the engagement. Thank you. And like the president said, you have my email. We can always interact. We can, yeah, that'll be good to hear from you if you need any specific question to your research, or probably you want to implement something, and then we can talk about the model in specific. So yeah.
Louise, do you have a question? Louise, I saw your hand up. Do you have a question? Okay, so maybe it was a mistake. Okay, so if we don't have any questions, Dr. Akinlade, we would really want to say a very, very big thank you to you on behalf of the executives and the whole graduate students wing, and even everyone that has participated in today's webinar. It's been very insightful, it's been very engaging, and we are so grateful for your time. We wouldn't have been able to have this as a success without you. So on behalf of the graduate student wing, we'd like to say a very big thank you to you. We know this is not the end of the engagement. We believe that this is the beginning of a beautiful relationship, and you would receive a lot of emails from us. Pardon us, but yeah, it will just show that you have really done a great job in sharing your work with us and also inspiring us and helping us to be better. So before I guess you already can see from the engagement in the chat that everybody has had an insightful time with you. So before we sign out, I would like, I don't know if you can, we can put on, you can go off
I
screen.
I can I can see a couple of my friends also here.
Oh,
That
okay,
was
that's
nice.
nice.
Yeah,
And
yeah.
please, we can also follow Dr. Akinlade on LinkedIn. So if you're not following him, I think I would encourage us all to follow him on LinkedIn. He's on LinkedIn. Let's follow him. Let's also follow the APBA Graduate Student Wing page on LinkedIn, because there will be more exciting webinars. And we are also gearing up for the APBA conference in Zimbabwe. So Doc, if you can go up sharing your screen so that we can have the pic, everybody can put, if you can put on your camera, please put on your camera so we can take a group picture with Doc. So if you can put on your camera, please, let's put on our camera so we can take a very beautiful shot. I'll be very grateful. It's good to see the faces behind the screen. It's very good to see. And please let us smile. I'm waiting for us all to join in. I really hope we get to see each other in Zimbabwe. So I'm encouraging us all to register. And if you're a student and you haven't submitted an abstract, please submit an abstract as well. We will be launching the African Journal at the APBA conference. So we'll be launching our own journal pretty soon at the APBA conference. And when you submit your abstract, you have a chance of being published in the journal. So please submit it. So if we are ready, I think we are. I'm going to take the first one and then I'll move on to the next screen and take. So please let us smile even as I do this. Thank you. So let me do it again. Please smile for the last time. Okay, let me move to the next screen. Okay, so yes, the next screen. Please smile. Okay, so thank you all very much. The webinar will be uploaded on the APBA YouTube channel. I've been able to speak with them. So the first one will be uploaded. I'm hoping by the end of the week that will be uploaded, and then this will also be uploaded. So kindly bear with us. The webinar would be up